Welcome to this explainer. You know, inside every single microscopic cell in your body, there's roughly 2 meters of tightly coiled DNA. It's this bustling, incredibly crowded city of interactions. And today, we're going to explore the toolkit scientists use to map that genome in real time. We are looking at the architecture of genomic interaction through a process called ChIP-Seq.
Okay, let's just dive right into it with the definition. ChIP-Seq stands for Chromatin Immunoprecipitation Sequencing. Yeah, I know, it's a total mouthful, but at its core, it's a really powerful lab technique. It's basically a way to track down exactly where specific proteins are interacting with our entire genome. Think of it like a GPS, but instead of finding cars on a map, we're locating vital proteins on the sprawling map of your DNA.
And I absolutely love this description because it brilliantly illustrates the whole concept. ChIP-Seq acts exactly like a camera. It captures a literal snapshot of where proteins, things like transcription factors or histones, are interacting with the genetic code at a precise, fleeting moment in time. It completely freezes the action, so we can actually study it. So, to grasp how this works, we're going to treat the workflow just like a photographer's journey. First, the genomic snapshot. Second, fixation and fragmentation. Third, immunoprecipitation and sequencing. And finally, reading the data peaks.
All right, Part 1, the genomic snapshot.
Moving right along to Part 2, fixation and fragmentation.
Okay, so this is where we actually get our hands dirty in the lab to freeze our genetic frame. This first critical phase operates a lot like a microscopic assembly line. We use 3 really strict sequential steps to prep the raw cellular material. We've got cross-linking, then lysis, and finally, chromatin fragmentation.
Step 1 is fixation, or cross-linking. Now, proteins are super dynamic, right? They're hopping on and off the DNA constantly. So to catch them in the act, scientists use a chemical fixative, usually something like formaldehyde, to act as a molecular glue. This creates strong, covalent bonds, basically locking the proteins tightly onto the exact DNA segments they happen to be touching right then. Boom, the cellular state is perfectly preserved.
But wait, there's a catch. All of that beautifully preserved material, it's still trapped inside the cell. So next, the cell membranes are lysed, which just means they're chemically busted wide open, releasing this cross-linked chromatin out into the open so the DNA can be cropped down to size.
Now for the crazy part. To shear these incredibly long strands of DNA down into manageable 200 to 600 base pair pieces, scientists have to get pretty aggressive. They literally blast the chromatin with high-frequency sound waves in a process called sonication, or they use specialized enzymes like micrococcal nucleus to digest it. This fragmentation creates the perfectly sized puzzle pieces we're going to need for the next step.
Which brings us to Part 3, immunoprecipitation and sequencing.
All right, so we've chopped up our genome into this cellular soup of fragments, and now we need to go fishing for our target. Immunoprecipitation sounds ridiculously complex, but it's really just an amazing microscopic rescue mission. First, an antibody designed to hunt down our specific protein is tossed in. It grabs the target, which, remember, is still glued to its DNA fragment. Then, magnetic or agarospeeds coated with protein A or G are dropped in to hook onto the antibodies. And finally, using an actual literal magnet or a centrifuge, scientists physically drag that whole complex straight out of the solution. All the other generic cell debris just washed away.
Once we've successfully hooked our target, honestly, we don't even need the protein or the antibody anymore. We just want the DNA instruction manual it was attached to. So we blast it with heat to chemically reverse those original crosslinks. Then a protease enzyme is introduced to just digest away all the proteins, leaving behind nothing but pure isolated DNA fragments.
And here is the absolutely crucial point. Those isolated fragments are then prepped into a genomic library. We use next-generation sequencing, or NGS, to read these fragments, translating them into millions of short sequence reads. We've officially digitized our biological snapshot.
But let's be real. Millions of tiny digital fragments don't exactly mean much on their own. It's like having a million puzzle pieces with no picture on the box. So to make sense of it all, these individual reads are fed into bioinformatics software. The computer takes the snippets and mathematically aligns them back to a complete reference genome.
And that leads us perfectly into Part 4, reading the data peaks.
When the software maps those reads back onto the reference genome, it's specifically hunting for peaks. These are dense clusters of overlapping sequence reads. We know they're significant because they pile up way higher than a control sample, one that wasn't sorted with our special antibody. Basically, a peak is the data screaming, hey, your target protein spent a ton of time sitting right here.
When we look at these read pileups, we're generally seeing two distinct shapes in the data: narrow, sharp peaks and broad peaks. And they tell us totally different stories about what the protein is actually up to.
If the software spits out a narrow or sharp peak, it's telling us something highly precise. It indicates a sequence-specific transcription factor binding site. Basically, the protein sat down on a very concise, tiny little snippet of DNA to do a highly targeted job, like flipping a single genetic switch.
On the flip side, if we see broad peaks, it means the interactions are smeared out. Broad peaks typically point to histone modifications, like H3K4ME3 or H3K27ME3. Since histones are the spools that DNA winds around, modifications to them tend to spread across much larger domains, sometimes covering entire gene bodies. It's a wider structural change, not just a tiny precise switch.
So by carefully manipulating this whole workflow, from freezing interactions with formaldehyde all the way to magnetically rescuing specific DNA sequences, ChIP-Seq gives us this totally unprecedented view into the mechanics of life. Knowing we can map these intricate interactions by literally freezing them in their tracks, it leaves us with an incredible question to ponder. What other biological mysteries can we solve now that we have the technology to freeze time inside our own genome? Thanks for joining me on this explainer, and keep questioning the incredible architecture of life.